I'm Dr. T and welcome to my office. So today let's talk DNA. Well, I should say let's talk nucleic acids, and to do that let's look at DNA because in many respects it's the simplest. Okay, so we're going to be using the deoxy variant of our nucleotides. So yeah, we've got these guys. We're going to line them up. They're going to be in a polymer. The way that they are connected is you take the nucleotide, the 3 phosphates on the 5' end. We're going to split 2 of those off. That's actually going to power the whole assembly process by splitting those off. That's kind of what they normally end up doing, is providing an energy source. Now, once you've done that, or in the process of doing that, really I should say, the remaining phosphate is then going to be connected both still to that 5' carbon, but also to the 3' carbon of the previous nucleotide. Now you're able to stitch this together. So you're going to have this pattern where you've got the ribose, and then it's going to be connected on the 3' carbon to a phosphate group, which is connected to the 5' carbon of the next ribose, which the 3' carbon of that ribose is connected, and so on and so forth. Okay, this is how you're going to connect both DNA and RNA. This is how your nucleic acids connect. The 2' position doesn't do anything, and that's why it's missing on the DNA, to make sure it doesn't do anything, because it can do this weird reaction where it breaks the phosphate connections in that backbone. And of course, you don't want it to do that. Okay, so you've got your connections. The way that these get assembled is they're going to pair up. And DNA you will find almost always as double-stranded. RNA you usually don't find double-stranded. It does exist, but those are usually in viruses, and your cells react accordingly, with extreme prejudice. But when you look at DNA, it's going to almost always have one strand, and that strand is going to have a complementary strand. And this is actually how it ends up replicating. So you're going to have a nucleotide, and if you have an adenosine, it's going to be paired up with a thymine in DNA. And then if you have a cytosine, it'll be paired up with a guanine. So you got this ATGC pairing. You'll notice this is a purine and a pyrimidine. So you have actually a total of 3 rings in your pair. They're connected by hydrogen bonds. The A and the T can do 2 hydrogen bonds, the C and the G can do 3, and because you're doing pyrimidine-purine pairing, enzymes can do a spell check by kind of patting down the DNA going, yep, it's all straight, it's not lumpy, it's not like it's got a poop that's sticking in. We got 2 of the single rings, like say a T and a C, nor do you have a case where you get like an A and a G where it's sticking out. So if it does this, then it's like, oops, I got a problem. Okay, so you've got your error checking in that approach. Additionally, what will hold this together, so the 2 strands being held together first by hydrogen bonding, and second, when you stack aromatic rings on top of each other, you actually get this interaction with intermolecular forces where they stack, and they like to stack on top of each other. So that will help hold your DNA together as well. Interesting enough, this is why aromatic compounds can be somewhat dangerous, because if you've got, say, leaves of the book, it's easy to slip something else in it. Think drying leaves. Well, certain aromatic compounds, like benzene itself, can slip in between there, and then when the DNA is being copied, that can create errors. Errors in DNA lead to broken things within the cell, and one of those things that can get broken are the machinery that prevents the cell from dividing like crazy and going rogue. Cells dividing like crazy and going rogue is what cancer is. Therefore, things that slip in between the nucleotide bases in DNA, third hand to describe this, those are very easily going to be carcinogens because those can mess up the DNA, and it makes things more error prone. You've got the ability to address problems, you can fix mistakes, but the more mistakes there are, the more likely one of them's not going to get fixed, kind of scenario. Okay, so that's DNA. DNA is used for data storage. It is basically your archival grade. You don't have much within your cell. Now, admittedly, the DNA strands are humongous. Human cell, you got like 6 foot of DNA. It's really tiny. It's wound up real tight. But that's all you have. You basically have 2 copies of your genome in DNA, and that's about all the DNA you really have. You don't really use much DNA. Most of the nucleotides that you use, those are going to be RNA, and I'll talk more about them later on because they really go into a lot of the machinery of how DNA takes that information from the DNA and makes it in the proteins that makes the cell, etc. But RNA, you can get that in the oligomer form, which generically is called small RNAs. And small RNAs come in a bunch of different varieties. These are used heavily for regulation. Basically, hey, if this matches your messenger RNA, which is a polymer form that allows you to transfer information. If this matches the messenger RNA, yeah, just get rid of the messenger RNA. Or it allows for modification. Like, okay, so if it matches here, then I need to do this, this, or this. Basically, post-production editing, what I'm going to do to this video here shortly. But on RNA. So you can use the RNA to regulate, you can use the RNA to defend, basically like a wanted poster. If you see this nucleic acid, destroy it, kind of thing. That's a fairly common defense in several different types of organisms. And then it can be used for, once again, proofreading or to assist proteins and whatnot. Now, RNA in its polymer form, where it's much longer. Because remember, the difference between oligomer and polymer is basically just size. Okay, so in the polymer form, RNA usually is not double-stranded. It does exist, but that's, as I mentioned earlier, mostly just in viruses and is usually treated very unkindly by cells because it's recognized as a threat. But this will exist in a single-stranded form, so basically just like DNA, except there's only one strand. This would be messenger RNA. Now, it's not just going to float around as one strand. It gets modified, it gets turned into kind of a loop shape, and this job's kind of like a photocopy. So you take the information from the DNA. The DNA is sequestered inside the nucleus. That way it's nice and protected. The messenger RNA is made in the nucleus and then shipped out into the cytoplasm or the endoplasmic reticulum, etc., and then that is used. That way it keeps the DNA nice and safe. Also, you can have things like ribozymes, which are going to be chemically active macromolecules, but unlike enzymes, which are made of proteins, they are these chemically active macromolecules made out of RNA. And then you'll get things like transfer RNA. So it gets used a lot of different places. We're really, in the last 20, 30 years, really starting to understand how important RNA was, or is. Back 30, 40 years ago, RNA was kind of a footnote, and proteins were the whole story. Now we realize that RNA is really putting a lot of the story into place, and it does a lot of different things. So with that said, have a wonderful day, and I'll see you in later videos.